So, hello everyone. Today we are going to discuss product formation kinetics. So, if you remember the last lecture we discussed structured kinetic models which gives the insight of cellular environment, and particularly we discuss these two models, compartmental models and model based on the cellular energetics and metabolism. So, compartmental model is based on this function, synthetic compartment and synthesis compartment, and the second one is based on the metabolism. So, today we are going to discuss product formation kinetics. So, similar to this previous microbial growth kinetics, first we will be discussing unstructured kinetic model, followed by that today we will discuss chemically structured kinetic models. So, unstructured kinetic model, as the name suggests, it considers cell as black box where substrate is converted to your biomass and it is also converted to your product. So, it did not consider the details of intercellular states. So, it is based on simple stoichiometry connection between product formation and substrate utilization or cell growth. As we have discussed during the stoichiometric balance for substrate utilization and product formation. Now, typically if it is unstructured model, so the rate of product formation is directly proportional to rate of biomass growth into a factor, that is y p by s. So, it says that you can write the rate of product formation either in terms of rate of biomass growth or in terms of rate of substrate utilization. So, why it is negative? Because substrate it typically gets depleted, and when it is the rate of substrate utilization, we have to multiply with y p by s into r f s minus r f p. Or when you are writing in terms of rate of biomass synthesis, then you have to write yield of y p upon x. And this typically holds good for type 1 fermentation. On one example, you can see the growth associated alcohol production in a batch culture. So, you just see the data here, A, B and C. A is the biomass, B is the product, your alcohol, and C is the substrate used. So, you just see particularly here this A and B, if they are following this parallel trend, and the moment biomass is reaching to the plateau, your substrate, your product is also reaching to the plateau. So, it says it is product formation is taking place during the growth phase and it is a growth associated product. Further, in the case of secondary metabolites, where the product formation takes place during stationary phase. So, for example, antibiotics, they are non-growth associated products. So, many secondary metabolites are non-growth associated product, and synthesis happens under appropriate conditions, particularly during stationary phase, and very minimal synthesis during this growth phase. So, in this case, the product formation is not proportional to the rate of biomass growth, rather it is proportional to the biomass concentration. So, here rate is proportional to the x, because this is directly dealing with the stationary phase. And we have previously discussed the characteristics of secondary metabolites. They vary greatly in their particularly chemical structure and biological activity. So, if you just see primary metabolites and cells energy charge, they are important modulator of reaction pathway leading to secondary metabolite products. And if you take example of antibiotics, so there are more than 600 antibiotics have been identified, and particularly this Bacillus subtilis and Streptomyces griseus, they produce almost 50 different types of antibiotics, 50 different antibiotics. So, if you just see this figure batch cultivation of antibiotic fermentation, let us see this A profile of your biomass and B profile of your product. So, if you see the A here, A has reached to the plateau phase, further it has entered to death phase as well, but if you see the region here to here, the product formation still continues. So, it says this product formation is non-growth associated, and during even stationary phase or even at the beginning of death phase as well, there is synthesis of product. So, what we have seen, the product may be growth associated or non-growth associated. In fact, in some cases there can be a mixed type, both growth as well as non-growth associated. So, what we have discussed, in the case of growth associated, the rate of product formation is proportional to the rate of biomass microbial growth or rate of substrate utilization. So, it says during the growth phase, simultaneous production of products along with biomass growth, and this is linked with primary metabolic pathway, and as we have discussed, as it is linked with this growth, so most of the production happens during the exponential phase. While in the case of non-growth associated, the rate of product formation is proportional to the biomass concentration, not with the growth rate, and mostly this production happens in the stationary phase. Now, considering in some cases there is a mixed growth as well as non-growth associated product formation. So, one model was developed by this Ludeking Pirate. It says rate of product formation is a function of biomass growth rate as well as biomass concentration. So, it says dP upon dt equals alpha dx upon dt. So, this dx upon dt is the rate of biomass growth. It means alpha, this term is representing for the growth associated, plus beta into x. So, it is directly proportional to biomass concentration. So, this term is representing from non-growth associated. In simply it can be written R Fp equals alpha into R Fx plus beta into x. So, one example for the mixed fermentation is the lactic acid fermentation by lactobacillus, and the product formation takes place during this growth as well as the stationary phase. So, typically if one can use this model and get the value of alpha and beta. For example, if it is a growth associated, then what will happen? Beta will turn out to be 0, then automatically this equation will be reduced to the growth associated equation. Now, how to estimate this parameter? The first thing, you have to find the specific growth rate and the x saturation value or the maximum value of x. So, for this this is simple logistic equation or even monad equation can be applied to get these two parameters. Then let us say during the stationary phase, it says dP upon dt equals alpha dx upon dt plus beta into x. So, if you only consider the stationary phase, this term can be neglected. So, we can find the parameter of beta as dP upon dt divided by x. Similarly, one can find the value of alpha during the growth phase, and once these alpha and beta is known, this substrate yield can also be calculated by simple mass balance, which we have discussed in the week 2. Now, let us see the kinetic data based on this mixed kinetic model. So, for ethanol production, alpha is 0.9 and beta is 0. It means it is a purely growth associated product. If you see the penicillin, very small value of alpha and large value of beta, it says this is a non growth associated production of secondary metabolites. Next, if you just see xanthan gum, it shows the mixed kinetics, where the value of alpha is 1.83 and beta is 0.155. So, you can see the both the values are present here. So, this is mixed kinetics, where both growth associated term 1.83 for the alpha and for the 1.155 for the beta is present here. Now, further what happens in what it has been observed, that during this production process, the product formation even can be complicated. It means there can be formation of more than one product at a time. So, for example, if I see this production of acetone and butanol by Clostridium acetobutylicum. So, it says in the initial period glucose is converted to acetic acid and butyric acid, and which subsequently are converted to acetone and butanol. So, it says that there can be synthesis of more than one product, and both the product may not follow the similar trend. They may, one may be growth associated, other may be non growth associated. Another example is product formation by cell may occur simultaneously with chemical reaction of products in media. For example, spontaneous hydrolysis of penicillin. So, for such cases the additional equations will be needed to exactly model the experimental data. So, considering the different behavior of product formation, Dendiofer classified the fermentation pattern as simple, simultaneous, consecutive and stepwise. Simple means these nutrients are converted into products in a fixed stoichiometry without accumulation of intermediates. Simultaneous means nutrients are converted to products in variable stoichiometry proportional. It is not fixed, it keeps changing, still without accumulation of intermediates. Next comes the consecutive. Here nutrients are converted to products and along with the accumulation of intermediates. And fourth one is the stepwise, which says the nutrients converted to first intermediates and then they are converted to the product. Here the selective conversion of product may also take place. It means all the intermediates may not be converted to the product. So, these were the product formation kinetics based on the unstructured kinetic model. Now, we are going to discuss chemically structured kinetic model. So, as we know the word structured means it is a, now we are talking the growth associated. So, one example for this is antibiotic production, and let us say this is inhibited by the high amount of phosphate. In fact, one can also take example of other secondary metabolites like alkaloids, which is produced by claviceps species. So, what it says, the chemically structured model are used to describe the formation of secondary metabolites in microbial culture, and these models incorporate chemical factors such as nutrient concentration, which affect both growth as well as the product synthesis. So, why we are talking about the particularly phosphate here in both the cases? It says because phosphate concentration plays a critical role in regulating the production. A high phosphate concentration inhibits the product formation, but at the same time the phosphate is required for the growth. So, what it says, an optimum phosphate level is required. So, just to see how we can model this kinetic data, first let us see the experimental data. So, all the points are the experimental data. So, we are presenting the 4 different initial phosphate concentration. There is no phosphate concentration 0.25, 1 gram per liter and 5 gram per liter. So, you just see Ta means total alkaloid, that is the secondary metabolite. So, if we just see compare the figure A and 1 and 2. So, in the presence of phosphate, the titer of the product has significantly increased. So, it says initially the phosphate is accelerating the product formation, but further if you just see compare figure 2 with 3 and 4, when we have increased the phosphate concentration to 1 gram per liter, that alkaloid concentration has significantly decreased, and it has been further decreased if I increase it further. So, it says that concentration is playing a dual role. So, what is happening? It is linked with the some internal mechanism, how it is regulating the product formation. So, when we go for kinetic model, so it will be done in the 4 steps. First, we are going to see the growth and extracellular phosphate balance. So, for the growth, we are writing this dx upon dt equals k1 1 minus e to the power minus k1 pi phosphate concentration to x minus k2 into x square. So, here the specific growth rate or the in first term is taken based on this Tesser equation, which we have discussed in the previous week, and second term k2 into x square is used to describe the rate of cell lysis. So, we are saying there is a growth as well as there is a cell lysis, and when we see the extracellular phosphate concentration, dp upon dt, this equals to minus k3 p upon p plus capital K2 x plus yp upon x plus pi K2 into x square. So, it says this the cell lysis releases the phosphate into medium, that is saying extracellular phosphate, and this is proportional to the cell mass phosphate content, that is the Ye P by x and intracellular phosphate concentration Pi. So, the rate of extracellular product formation is proportional to Yp by x plus Pi and in addition the first term describes the active transport of phosphate into cell with the saturation kinetics. So, if you just see, this is a typical equation for the saturation kinetics. After certain concentration it will reach to the plateau. Now, other two part is intracellular phosphates as well as the product formation. So, in the case of intracellular phosphate DPI, we are writing DPI means intracellular phosphate. So, it come out to be DPI upon dt, this is the K3 P upon P plus K2 minus K1 yp by x plus pi 1 minus e to the power minus k1 into pi. So, what it says, what is there in rate of intracellular product formation? It should be the difference between the rate of phosphate transport inside the cell and rate of phosphate incorporated in the cell material. So, if you do the difference, particularly if you look how much phosphate is going to the cell material plus how much phosphate is transported into the cell. So, what is the transported? That is K3 into P upon P plus K2 minus how much is internalized into the cell, that is K1 yp upon x plus pi into 1 minus e to the power minus k1 into pi. So, it is very simple difference between the phosphate transport in the cell and the rate of phosphate incorporation in the cell material. Similarly, if we just see the last one, the fourth equation, rate of product formation, it says dA upon dt is equals to a constant K4 K3x upon K3 plus pi square. So, if you remember this term pi square, this we have previously discussed along with the substrate inhibition kinetics, right. So, the product formation kinetic model includes the inhibition by the phosphate. So, basically what it is doing, it is repressing the particular phosphatase enzyme, in that way it is inhibiting the product formation kinetics. Now, based on these four equations, the experimental data were modeled, and if you see in the same figure here, the solid lines are the predicted data. So, if you see this, the models have quite this nicely this validated the rather fitted to the experimental data. So, it says now we should be able to calculate those kinetic parameters. So, what these are the value, this K1, K2, K3, K4 as well as yp by x, K1, K2 and K3. So, these are the model parameters which were estimated after fitting the experimental data, and what it predicted is this optimal phosphate concentration is between 0 and 0.25, it came out to be 0.17 to maximize the total alkaloid yield in a batch culture. Further, it also indicated there is no further gain in product with increase in the further phosphate concentration. So, what additional information we are getting here is this model guides to the particularly media formulation, and this also helps in the feeding strategy, particularly when we will discussing for the fed batch culture. So, for such fermentation one can go for the fed batch culture, because you are observing the inhibition by the nutrients as well as the optimized condition of nutrient is required here. So, with this I will stop here for today's lecture, and in the next lecture again we will continue this product formation kinetics. There are two more models. First is based on the genetically structured model, and next one we will be discussing the product formation kinetics by filamentous organisms. Thank you so much.